Great. I think we'll make a start. The numbers have started to stabilise. There'll still be people entering the room as we start the webinar. But welcome and thank you for joining us today for our Health Matters webinar at St Vincent's Institute at SVI. My name is Professor Natalie Sims. I'm the Deputy Director of St Vincent's Institute and Head of our Bone Cell Biology and Disease Unit. And I'm really pleased to welcome you here. Before we begin, I'd like to acknowledge the traditional custodians of the various lands on which we're on today. St Vincent's Institute is in Fitzroy. We're on the lands of the Wurundjeri people, and I'd like to acknowledge the Wurundjeri people as the traditional custodians of the land on which our institute is located, and pay my respects to their elders past and present, and extend that respect to any Aboriginal or Torres Strait Islanders who are participating in the webinar today. Today, our focus on the webinar, this is our fourth webinar for the year and our last webinar for 2022. We're going to be focusing on dementia. And today you're going to hear from Chris, who's a dementia advocate, and followed by our Professor Michael Parker, who does research on dementia. So he'll talk about it from a scientific perspective. After both of their presentations, I'll interview Chris and we'll hear from Michael. We'll have a short question and answer session where you'll have the opportunity to have your questions answered, both by Chris and by Michael, so those two perspectives. You'll see that there's a Q&A button at the bottom of your screen, and that's where you can type in your questions, and at the end of the session I'll get to those. So feel free to put in your questions at any time when there's something that you would like to ask, and I will get to them at the end of the talk. We'll just be using the question and answer feature today rather than the chat or the raise hands options. So please put your questions in there. Right, so that's all of the housekeeping out of the way. I'm now going to introduce Chris, who is going to be talking with me today. Chris was diagnosed with Alzheimer's just in the last couple of years, and he's an advocate with Dementia Australia. And we're really grateful to you, Chris, for sharing your experience today. And we're going to begin the webinar by me asking Chris a few questions and talking about his experiences and what he can tell us about being a patient. So Chris, welcome. Thank you so much for joining us.
Thank you.
Let's start by talking about when you were diagnosed. And could you tell us about when you were diagnosed with dementia and what were the symptoms that you were experiencing when this happened?
It was mostly just forgetting lots and lots of things. I was a bit like I work as a sorry.
It's okay. Take your time.
Builder. I was a builder. I was not anymore. And I just noticed that I was forgetting things, words. I went to the psychologist and he had some other little things that I should do, but that was pretty quite useful, not useful. And I went to the hospital instead and started that process of to find out what's actually had happened. And now I've done that, and now I'm here.
Yeah, and can you tell us something about what your thoughts were when you first got that diagnosis? How did you react to that?
I didn't want to know anything about it really. I just didn't really want to face up to it for a while. In the end, I had to, to go to see other people and see, okay, what can I do? What's available? Which wasn't a lot, but yeah, I don't know. That's the normal way it is, but and so I haven't really done much for it since then, because some of the ones that are getting trialled, I haven't been able to do it because I separated from my partner. So see someone in that situation. Does that make sense?
Yes. Yeah.
It'd be good. if I could join that as well, you know. But I don't know how the processing would be.
Yeah, yeah. So the extra support that's needed to participate in those things is an issue.
Yeah.
Yeah. Before your diagnosis, what did you know about dementia?
Nothing really.
Yeah.
I just didn't know the name and didn't really know deeply what it was about.
Mm-hmm. Mm-hmm. So it's been very much a learning experience for you since your diagnosis.
Yeah. Well, it's taken over my life, really. You know, it's all about what's happening, you know, with my mind and how I'm dealing with it.
Yes. Yes.
And just basic things, words and stuff like that. It's okay sometimes, and other times it's not really, you know, it's quite changed. But this year though has been probably a bit better, I think.
Mm-hmm.
So you go through up and down with this because there's not much that we've been able to do anything. Where does that leave you? You know?
Yes. Yeah.
But lots of people in that situation.
Yes. Is this? I'm going off script here a bit, but I'm intrigued to know: is this year different because was COVID a more difficult period because of isolation there?
I haven't had anything from that.
Yeah.
I've been lucky.
Good. Good.
Yeah, it's good.
And are you on any therapy for?
Just a basic one, which is not the Adrian. Do you know that?
I don't, but Michael probably does because it's not my field.
It seems to this year's been. It seems probably been good with it. I think it's helped a little.
Yes. Yeah.
I'm probably just trying to. I mean, I work. I look after myself and food and all that kind of stuff, and probably just getting even better at that is what's been good. You know, some really good different food to eat and
Yeah.
yeah.
And what about hobbies and interests that you have? Have you been able to maintain those, or have you picked up new hobbies?
I've never had hobbies.
Yeah.
I haven't got any now. People they want to go into the men's shed, but I really, I was a builder for 20 years.
Yes.
I don't really feel like that's doing now. I mean, I can go and get a little bit of work for friends and stuff, just a little bit, and that's what I've been trying to do a little bit more. And not so much to get money or anything like that, just to be around other people during the day. You know, it's because when it's like this, when no one's here, it's it's quite not lonely, but it's just there's nothing really happening. You know.
Yeah.
But it's been, I think it's been a better year this year.
Yes. Yeah.
But it's up and down with this. Because it's it's not an easy track to do. That's what I've found. And most people don't understand very much at all. I mean, I can understand that for sure. There's more information about it now, which is good.
Yeah. While we're thinking about information, have you got any pearls of wisdom, any messages, any advice for others who might be going through the same experience, or who have family members going through the same experience who might be on the call today?
Yeah, what was it? So what was that question now?
Yeah, so any advice that you could give to people who might be going through a similar experience to you, who've just had a diagnosis, or for their family members, advice that might be helpful to them from your experience?
There's not a really big amount of things you can do, really. I mean, what can you do? What can you get from, say, this basic sort of thing? I don't really know much about it, but I don't know what is there for but yeah, what is there for dementia to change it?
Right.
I mean, it might be close to happening. I don't know. These other people might have a better idea.
Yeah. Well, I'm sure Michael's going to tell us something about what things are around the corner for dementia at the moment.
Yeah.
Yes.
Yeah. Yeah. Trying to stay with friends and all that. That's really important.
Mm-hmm.
That's work. You can't sort of go and hang around with your friends while they're working and you're.
Yeah. But building up those whatever friendship networks you can, and what you've mentioned about the men's shed sounds like something that's been quite helpful as well.
I haven't done the men's bed. Hold it. Men's shed.
Men's shed.
They put there's one here where I live,
Mm-hmm.
but that's really small, and that's getting to make it much bigger next year. But I haven't really been that interested in that, because it's okay to I don't know. I haven't been. There's a few people who've been putting it on me to get in there and do something. But, the one down at Alzheimer, he's got like 250 waiting to get in there themselves.
Oh gosh.
It's amazing how many people, mostly older men.
Yeah.
It's interesting.
And I guess my last question for you is what's your hope for the future?
Someone that, of course,
Yeah.
A cure.
Yeah, absolutely. Absolutely.
This can't. I mean, you could change aspects of your life or that, but it doesn't really change what's happening with the dementia.
Yes.
It's almost not useless, but it has to be something that's big.
Yeah.
Big thing that's happening all over the world. How many people are sick?
Yes. Yeah. Chris, thank you. Thank you so much for sharing part of your story here with us. What we're going to do next is Michael Parker is going to give us an update on the research that's happening on dementia at the moment. I think he's going to give us a bit of a historical perspective and then talk about the sort of work that is happening around the world and that his team is doing as well. So again, thank you, Chris. Thank you so much. And Chris is going to stay on the line. And if you have any questions for Chris, please do put them in the Q&A and we'll get back to them after Michael's part of the talk. So thank you, Chris. I'll now introduce Michael Parker, Professor Michael Parker, who's head of the Structural Biology Unit here at St Vincent's Institute at SVI. And Michael is also director of the Bio21 Institute at the University of Melbourne. Michael's an NHMRC Leadership Fellow in the Department of Biochemistry and Pharmacology at Bio21. Michael did his Bachelor of Science degree at ANU in Canberra, followed by a DPhil in protein crystallography from Oxford University. Michael came back to Australia to re-establish a structural biology lab at SVI in 1991. Michael has been very closely associated with structure-based drug discovery. Don't worry if you don't know what that means. He's going to talk you through it, I'm sure. And he's been involved in that for almost 20 years. He was head of Biota's Structural Biology Lab and head of the Structural Biology Node of the Cooperative Research Centre for Cancer Therapeutics until 2014. His work on dementia drug discovery, which has been for more than 20 years, has included collaborations with major pharmaceutical companies like Merck, Servier, and Janssen Pharmaceuticals. Michael is held in very high esteem around Australia and around the world for his research. He's published more than 300 papers and his work has been recognised with many awards, and two recent highlights are when he was elected a Fellow of the Australian Academy of Science in 2010 and a Fellow of the Australian Academy of Health and Medical Sciences in 2015. Michael, I can see you've got your slides up, so over to you. Please give us an update. Thanks.
Okay, thanks very much. And thanks for the invitation. Perhaps I should start off by saying that in terms of acknowledgement of country, would like to add the fact that our Indigenous people were the first scientists in Australia, and I think we've got a lot to learn from them. So with this talk, it's going to probably start off in a slight negative way because of the history of the disease, but there is some hope, some good hope, I think, which you'll see at the end of my presentation. So I think Chris has done a terrific job in sort of painting the picture, which is a bit of an unintentional pun, because I want to first start off by talking about an American painter called Williamuter Nolan, who was diagnosed with Alzheimer's disease in 1996 at the age of 61, and he did pass away when he was 73. So he was a portrait artist, and when he discovered that he was diagnosed with Alzheimer's disease, he started doing self-portraits, and there's some self-portraits up there, and you can see that over time they become more and more abstract, and that's because he was losing his identity. So a dreadful disease, but what about the history of it? So it was actually diagnosed as a disease way back in the early 1900s by a German, Alzheimer, and hence the name of the disease. And it's actually the first neurodegenerative disease characterized, and it's part of what's called dementia, which I'll talk about in a minute. So the way he found this is he had this patient that came to him, and you'll see the patient's photo down below. Only 51 years old, and in those days, the treatment, so-called, was to go to an asylum. But you can see some of the symptoms of patients who get this disease. So we've already heard about short-term memory loss, changes in personality, and eventually people will pass away. So dementia. So actually, the Alzheimer's disease and dementia is used interchangeably quite a lot. Dementia actually is a description of a bunch of sort of related diseases, which are shown here, but you can see Alzheimer's is perhaps 70% of the total. Each one of these diseases, they're caused by a different protein, and yet the result is quite similar in the end. And Chris has already alluded to this, but it's really is a major disease both around the world and in Australia. You can see in the graph there that Alzheimer's, Parkinson's, and so on is the second top killer in Australia. And then I've got some worldwide stats there, but if you're particularly interested in Australia, so at the moment we've got nearly 500,000 people in Australia that have dementia. And obviously the majority would be Alzheimer's. But very importantly, and with Chris today as Kate, is that there's actually 1.6 million carers. So it is affecting a lot of people. And the scary number, if nothing is done about it, is by about 2058, there will be over $1.1 million people with dementia. And the cost is tremendous. So in 2016, it was calculated, cost the Australian economy $14.25 billion. So it's certainly something we really want to find at least a treatment for. So when Dr. Alzheimer was looking at his patient, the patient passed away, and looked at her brain. This is her brain. It's a typical Alzheimer's brain. You can see a healthy brain, and the Alzheimer's brain on death is quite atrophied. And what Alzheimer did is he looked at slices of the brain, and he saw what's called pathologies, two things that you don't normally see. So pathologies is something to do with the disease. And one are these sort of circular type features here, plaques. And another sort of feature that he saw was tangles. And what we know these days, and took quite a few decades to find this out, that the plaques consist of a protein, a small protein, a peptide called A beta. and the tangles consist of another protein called tau. So the huge debate and confusion over many, many years in terms of research into Alzheimer's is, what is the cause of the disease? Is it the plaque? Is it the tangles? Or are they just an end pathology and something else causes the disease? As an aside, one of the things that's really frustrated the field and has only been solved in the last few years because it's been seen to be so important is actually diagnosis. So typically the diagnosis has been, as you saw from Dr. Alzheimer's time, autopsy, biopsy, and up until relatively recently is actually the patient actually coming forward to a GP, say, presenting with symptoms and then having a clinical assessment. And that sort of assessment is not foolproof. As you can see, there's a number of dementias. It's very important to know which dementia you've got. But what we've got now is a new technique, which is, I think, the gold standard, which is called PET diagnosis. So you go into a machine, you get a scan of your brain, and there are special radio tracers you can add, and they can light up. So in a healthy brain, that's what you expect. But in an Alzheimer's brain, you see a lot of the brain is lighting up, and that's this A beta peptide that the trace was looking at. So we're seeing where the A beta peptide is, and you can see there's a lot of it in the form of plaques and so on. So this is the scary bit and a really important bit, and there's another thing that's really frustrated trying to find a treatment. So what I've talked about so far is the symptoms. So if we look down the bottom there, generally, and you'll see some exceptions in a minute, but generally people get aware of symptoms in their mid-50s onwards. But unfortunately, the plaques and tangles I've been talking about, they're actually appearing a lot earlier. And we know that because of this sort of advanced diagnosis sort of method, that those sort of events are happening in your sort of 20s and 30s. And you'll see here, there is some where you actually have genetic mutations I'm going to talk about in a minute. So for quite a few of the listeners here, they may already have the disease or the start of the disease, and so we do need to find urgent treatments. One of the other really frustrating things at the moment, which I'll get to at the end too, is when is the best time to treat? Well, it probably makes sense you'd treat when you start getting these modifications occurring in your 20s and 30s. And is it too late if you start treating people in their 60s and 70s? So we'll get back to that comment in a little bit later. Now, just a little bit of biochemistry, because it's very important to understand where we are. So I've talked a lot about the A beta peptide, which is shown as a cartoon here in red, a red rod, the A beta peptide. That's actually a small, as I've said, is a small protein, and we call that a peptide. And in the 1980s, its chemical composition was worked out. And a very important part of that is with a protein, it's made up of amino acids in a sort of string of amino acids. And the technology to sequence that was actually done by what's called Edmund sequencing. Now, Pierre Edmund was the first director of St Vincent's Institute, and his automated sequencer you can still see in the ground floor area of our institute if you're interested. But the impact of that has been tremendous and certainly impacted our early understanding of Alzheimer's disease. So once we knew the chemical sequence of this peptide, then we were able to use molecular biology techniques to find out, well, where does this peptide come from? And it turns out it comes from a much larger protein, which is shown in the middle here, and it's called amyloid precursor protein for pretty obvious reasons. This is a protein that's the precursor to form the amyloid, which is A beta makes the amyloid. So how does the A beta get released from this big protein? Well, there's a certain type of protein in the human body called proteases, which just chop up other proteins. And the ones that chop up APP, or amyloid precursor protein, one of them is called beta secretase or base. The other one's called gamma secretase. Very importantly, with this gamma secretase, it actually consists of a number of proteins called presenilin. So this is where we get to see the genetic side and very importantly, why the field has got so confused. So there's a hereditary form of Alzheimer's disease. There's not that many people have the hereditary form, but what we do know for those who do have the mutations in their genes, they occur in either the amyloid precursor protein itself, about 15% of the time, but most of the mutations occur in the one of those two proteases, the presenilins of the gamma secretase. So you can see there's an enormous number of mutations that occurred. The other thing we know is that all these mutations or changes in the amino acid sequence occur around where APP gets cleaved or by the protein or enzyme that cleaves them. There's you may have heard of a very dreadful disease called Down syndrome, and the majority of Down syndrome is caused by a gene in the body that's triplicated. It's chromosome 21, and in that chromosome is amyloid precursor protein. So it's actually repeated. It's a lot more is made in Down's patients than others, and Down's patients don't live so long, and they do develop symptoms that look like Alzheimer's disease. Even more interesting, some years ago in Iceland, there was a study of people there because they didn't seem to get Alzheimer's very much. And there's a particular mutation, again, near the cleavage site, which actually was protective. So again, suggesting there's something about APP in the disease. Now, there is an early onset Alzheimer's, so you can get that in your teens. It's quite rare, but again, mutations, particularly one of these presenilins, are a cause of that, or at least associated with that. And maybe of interest, if you ever see clinical trials of AD drugs, Alzheimer's drugs, quite often they'll talk about patients in Colombia. So it turns out that some of the largest families with this hereditary type of Alzheimer's disease live in Colombia. So there's a lot of clinical trials done there. Again, many of these families have mutations in the presenilin. So I think the vast majority of scientists around the world feel that at least the hereditary form of Alzheimer's disease strongly supports the relationship of this peptide and these amyloids in the brain that's causing the disease. The problem is it's only 0.3% of all dementias. So there's been this big argument is, can you say if A beta is responsible for the disease for familial hereditary forms of AD, is it true for all other forms of AD? And there's been that ongoing debate, which has caused a lot of fraction in the community. One thing I will say is if you do look at the brains of people that suffer from early onset and look at those that suffer from late onset, in your 20s versus in your 50s, they do look very, very similar. So I have to just mention a little bit of my work as I go along, and Natalie sort of introduced some of that. The specific sort of title or sort of expertise I've got is something called protein crystallography, and the aim of protein crystallography is to try and get atomic resolution views of proteins. So it's like a very powerful microscope looking at protein molecules, which are very, very small. The first step we do is we form crystals, which act like amplifiers because there's lots of protein molecules within the crystal. We shoot X-rays, a powerful source of X-rays. So this is the Australian synchrotron, which is a source of very intense X-rays that is located at Clayton. Then we can shine X-rays at our crystals, and we get these patterns. They're called diffraction patterns. and then we convert them back into atomic structures. One thing I really wanted to mention here is that the method of being able to go from these spots here to the atomic structure was actually developed by an Australian. His name was Lawrence Bragg. He was born in Adelaide and went to Adelaide Uni, and then went to the UK with his father. In 1915, or the early 1900s, the Braggs, father and son, got a Nobel Prize. So Lawrence is our first Australian Nobel Prize winner for developing a very simple mathematical equation that will convert that into this. If you're wondering what this little structure here is and what it all means, this is actually a molecule that's found within a protein. The first protein structures that were solved had this molecule in them. It's called heme. And in the center of the heme, the brown ball there, is an iron atom. And bound to it is two red atoms there, two balls there. That's oxygen. So this chemical picks up oxygen, and then it gets transported through our bodies, so it gives oxygen to our cells. So when we breathe in oxygen, this protein called myoglobin hands it on to hemoglobin, and our cells need oxygen to survive. So I've shown you what the cartoon form of amyloid precursor protein looks like, but this is a more complicated sort of picture using crystallography. The actual atoms you can see is these sort of sticks, and where the sticks are the chemical bonds, and they join where the atoms are. Protein structures are really complicated, so we tend to simplify them, and this is what you'll see as we go on. So we have these beautiful sort of ribbon-like representations of the way the protein curls around. I think maybe the takeaway here is that the very simple view of what proteins look like, what you might find on the web, actually hides things that are very complex, but wonderful actually to look at and try to understand. So a lot of this is crystal structures that we've done, and what they told us is that this amyloid precursor protein has properties like promoting neurite outgrowth, transport metal ions, and we all do this by we get our structures, we compare them to other structures around the world, and then we can infer what the function of the proteins can be. It's very important to understand this protein because, as you've seen, it's pretty central to the disease, understanding what it does, and if we have drugs that might interfere with it, is that a good or a bad thing? I'm hoping that if you just focus on the picture on the right, it would be sort of familiar. If you look at the shape, this Y-shaped molecule, this is the shape of an antibody. And during COVID, I'm sure you've seen on television lots of pictures of these Y-shaped molecules attacking the coronavirus, particularly the spike protein. And that Y shape came from crystallographic structures of antibodies. Just focusing on this this picture here, this antibody, this is one of the antibodies that have been tested in clinical trials, crenezumab, attacks the A-beta peptide, and the A-beta peptide can kill neurons. These are the brain cells of our body that communicate through electrical and chemical signals, and the networks that neurons form are important for memories. But also shown here is these little star-shaped things called microglia. These are the immune cells of the brain, so very important to try and protect us. The other thing I wanted to point out here is that we're not too far away from blood vessels in the brain. And so one of the real big challenges of developing drugs that need to get into the brain is what's called the blood-brain barrier. So there's a lot of control of what can get across that blood-brain barrier. So we've got to be able to develop drugs with properties that can go across, and that's not easy. Again, a little bit of structural biology, but it's so important because it actually says why there's been a lot of trouble coming up with treatments. So what I'm showing here in this sort of twirly sort of diagram at the top is what the A beta peptide will look like when it's sitting in the cell membrane as part of amyloid precursor protein. But once APP gets cleaved, and this gets free from the membrane, it adopts another sort of structure shown by these arrows here, cartoon sort of picture of what's called beta sheet. This is a really horrible peptide to work with. It's a really sticky peptide, not only in a test tube, but also in the body. So it wants to actually get together with other A beta peptides and forms what we call oligomers, which are just a collection of the A beta peptides. They will assemble into what's called a protofibril, then fibrils, and then actually the plaques that we see in the brain. Very importantly, it was shown probably about 15 years ago now, 10, 15 years ago, that these oligomers here are really toxic to cells, to neurons, etc. You'll remember from the beginning, I talked about two different proteins. There's A beta and tau. So there's been an argument for a long time, which one is the most important one? In the last decade, it's been shown actually that what comes first is the A beta being produced, and that actually leads to a form of the tau that's toxic to the body. So when you hear about what's the best thing to target, it actually turns out there's companies developing drugs against the A beta. There are also companies developing drugs against tau. And amazingly, what we think, but this is still not conclusive, that the plaques themselves are actually protective. It's a way that the brain tries to get that A beta out of the liquid environment in the brain and try to make it solid so it doesn't cause any problems. So I'll quickly go through as time is running along. I just wanted to say this is what we've got here is an atomic resolution picture of A beta. You can see the atoms there, and companies have been trying to target the A beta peptide because they've been thinking that's causing the disease. Antibodies aren't big enough to attack the whole peptide. So there's some companies that have been are targeting one end, middle, and at the beginning end here. Our own group have been actually looking at some of these antibodies, how they work, because our hope is one of these antibodies would work. If we knew which one worked, then our structure would tell us how to make even better antibodies. It turns out what looks the most promising antibodies are one that target the left side of the picture there. So this slide is actually pretty recent, probably about a year old, and it was a very depressing time in the Alzheimer's field because quite a few people said this was showing that the amyloid hypothesis, the A beta hypothesis, is not the cause of the disease. There's a whole bunch of antibodies there. There's also drugs that target some of those proteases there, and enormous cost in trying to develop these. I just put the stars there because these are the ones that we've worked on from a structural point of view. So that's where we were about a year ago. So there is some good news coming up. But I just wanted to decide, this is something that Chris brought up, is that, what causes disease and what can you do about it? And there's a whole bunch of things, factors down here. I've already talked about age. There is some discussion about lifestyle. I've already talked about genetics. And this is what makes Alzheimer's so complicated, because it's not just neurodegeneration, but there's all other things that occur because of that. And I'm going to talk about taking advantage of immunity in the microglial cells in a second. But I just want to have one little tangent, which I think far from proven, but I think it might explain what one reason why people get Alzheimer's disease. We've already heard one, which is the genetics, the hereditary form. And this is a surprise. It sounds pretty out there, but in the last few years, we've started to see papers that show viruses associated with dementia. And pretty interesting here also is that the A beta peptide actually acts as an antibiotic. It can kill bacteria. So it's something I've always said to my own group: is A-beta good or bad? And I think that production of that A-beta peptide is probably a defense mechanism. It's just that when it gets out of control, then the microglial cells can't cope. They've taken it out of the brain. I've got a little sort of a little bit of text on the right-hand side there. It might sound wacky, but there was a huge leap in understanding another disease of the central nervous system, multiple sclerosis, that came out earlier this year, finding that there's a virus connection with it. And now we're starting to seriously look at some brain diseases in terms of infection. And obviously, observations like that might lead to some treatments. So very quickly, in the little time I've got left, a technique which I've named down the bottom there, genome-wide association studies, can be carried out on patients that suffer from Alzheimer's disease, and ask the question, well, what genes are mutated in people who've got the disease? And we've said the obvious one of amyloid precursor protein, the proteases, but it turns out there's quite a lot of proteins in microglia, which are the brain immune cells. So there's been a lot of attention being focused on them. As I say in this slide, the microglia, which are right in the center here, are the garbage collectors of the cell. They clean up all sorts of stuff. I have proteases inside the microglia that chew up protein-like things, and then they get ejected from the brain. So the trouble with microglia when they get excited by things like A-beta is they secrete some molecules to try to combat the virus or the bacteria or the A-beta, and these are called cytokines. And some of you may have heard in the media, for example, with COVID, something about cytokine storms. With influenza, some young people sometimes get more badly affected than older people. And that's again because the cytokine, they're very healthy, their immune systems are very healthy, and they produce lots of cytokines, which if they're produced for too long a time, they can do a lot of damage to the brain. We've been doing our grants, funding for our work, focus on a bunch of different approaches. For Dementia Australia Research Foundation, we're looking at an antibody that's a very small version of the antibodies that I've talked about today. The smaller versions, and we've got data for this, can actually get across the blood-brain barrier a lot easier. We're looking at molecules on the surface of microglia to try to make the microglia more active in chewing up the A-beta peptide. And we're also involved in developing better diagnostics, radio tracers for the sorts of microglia that tend to be activated and cause problems. So I promised, this is the second last slide. I promised that there's some good news, and there's some really good news, but importantly, this news has not been peer reviewed yet. This is a release from a drug company, as Eisai and Biogen, and it concerns one particular antibody called lecanemab. Now, I've talked about the different sorts of stages or forms of the A-beta peptide. You can see there's a variety here, and I did say these earlier forms seem to be toxic. What this particular antibody does, it attacks a particular form of A-beta. Now, there's one that is out in the clinic, which controversial. It doesn't. We're not quite sure whether it really works, and that's aducanumab, and it attacks another form of A-beta, which we're starting to think is the wrong form. So the endpoints for this phase 3 clinical trial, the cognitive decline was reduced by 27%, which is quite good. It's an antibody, so it has to be injected. You can't take it as a pill. And what they found is they didn't find much effects after 12 months, but after 18 months, then they found the effects, which seemed to be quite convincing. You do have to use a very high dose. It's going to be very expensive. There was some side effects, but no obvious symptoms from those side effects. So why do we need new drugs with this very promising drug, which has been given accelerated accelerated progression through the FDA. So if all goes well, it might get approved next year. The reason why we want more drugs, well, it's not perfect. It's we want to reduce cognitive decline by a lot more than 27%. We don't want to use very high doses. It's not only expensive, but it can cause these side effects. We don't want to be taking a drug that you have to take twice monthly for quite a long time. And we still don't know yet, would you have to take it for life? There is some evidence on this antibodies that you might have to in terms of the cognitive decline. And the other thing too, as I've said, there's this other protein, tau. And is tau is causing its own problems. So maybe we need more than one drug. So I think things are looking really exciting, really promising. This antibody is supporting the amyloid hypothesis, but more work needs to be done there. So I want to end off thanking my lab, and the members are listed there. I think if you sort of first focus on the central part of this diagram, it says something very important. With Alzheimer's disease, where there's so much controversy, and we really don't know enough about the disease being a brain disease, it's a very high-risk area to fund, and this is where philanthropy's been fantastic. Philanthropy is prepared to take the high risks, which the more established bodies like on the right side, the National Health and Medical Research Council, or industry, which we've partnered with, they are a lot less risk averse, and so it's more difficult to get money. Your discoveries have really got to be very advanced before they start thinking of funding. The final comment I'd like to make, which you might think is a little bit strange, I've got here the Australian Cancer Research Foundation. Now they've given St Vincent's quite a lot of money over the years for equipment for looking at cancer, but they do allow the equipment to be used on other diseases. Obviously the focus should be on cancer. And some of the equipment we've used in our research has been supported by the Australian Cancer Research Foundation. One of the great things about SVI is we're not focused on a single disease. We've got expertise in all sorts of diseases, and that's good because we don't have those boundaries of that high focus on one disease and ignoring the discoveries that happen elsewhere. Some of the proteins we work on actually targets in cancer. Proteins that genes that get mutations and the resultant proteins can cause different types of diseases. And so having that sort of whole of sort of a number of diseases is a very powerful thing that SVI does. So I'll end off there, and thank you for your time, and happy to answer questions.
Great. Thank you so much, Michael, for that presentation, and particularly for ending on such a hopeful note about the potential new therapies that are coming through in drug development at the moment. It's time now to hand over to our audience for some questions that we can answer. Please, if you've got any questions, please put them in the Q&A box at the bottom of your screen, and they can be questions either to Michael or to Chris, so that you can hear more about the science or more about the experience of being a patient with dementia. We've already got a couple of questions in there, you know, please just feel free to put your questions in, and we'll gradually work through them. There's plenty of time. I might actually start with a question which has come through for Michael, and this is from Mitch. I'll take it a little bit slowly because it's a little bit complicated. So Mitch has asked really two questions, and the first one is how common among scientists is the view that the plaque might be protective, so the amyloid plaque is protective. And if that is the case, then what are the common treatment targets that are not focused on the plaque, if that is actually protective?
Yeah, I think the really important point I want to make with the presentation is there's these different forms of A beta. And the plaque is one form, which is that end form. And the argument would be if A beta is a good target, we need to think about which of those forms we need to target. And that very positive looking antibody is targeting an earlier form, not the plaque form. In terms of the plaque itself, like everything in Alzheimer's, there's disputes. you may hear that some people would die who seem to be healthy just before. And when you look at their brains, they do have amyloid in there. So, so some people say, well, amyloid's not to do with it. Others will say, well, if you look at it closely, it's a different sort of amyloid. So the idea that plaques are protective is an area of controversy, but I think a large number of scientists do believe it's protective. So, but that doesn't mean A-beta is a bad target. It's those earlier forms where you want to target. In terms of other targets, we saw it on the slide, the proteases, that's to do with the A-beta, I guess. There's tau, which is downstream of that. There's a lot of work in neuroinflammation now, which it doesn't matter what the cause. So, and that's one of the reasons we work on the microglia. It doesn't matter what causes the disease. The neuroinflammation can cause a lot of damage to the brain. It can kill neurons and so on. So ways to approach this is to try to stop the microglia from getting into a bad form, which causes all this damage. The other one is the cytokine storm, and there's actually antibodies to cope with that too. So there's a whole bunch of approaches.
Yeah, thanks, Michael. The next question I've got is for Chris. Chris, you talked about how this year has been better for you than last year. And Nicole has asked, do you have any ideas why this year has been better for you?
Maybe I felt a bit more bright.
Yeah.
I don't know. I don't know that. Yeah, I can't answer. I don't know. I have to. Sorry.
No, that's fine. Sometimes things are just better, and sometimes they're not. That's okay if there's not a particular reason that you know of. Thanks. Yeah. We've got a question, and this one is more for Michael. This is from Claire, who's a registered nurse. Claire is asking, since a diagnosis isn't made until symptoms are already evident, is it worthwhile taking or developing something that could be a preventative treatment?
Yeah, look, I think the aim of the field is first to just get a treatment that reduces the decline. Then the next aim would be to be able to be preventative. The trouble with the antibodies, they're biological molecules, they're proteins, they're very expensive. And so it's difficult from the economics point of view to understand how they will work. But if you can, I've always said with Alzheimer's, if you can find the drug that treats the disease, you're going to find out what causes the disease, and then you'll get to cheaper drugs. Sorry, I'm just trying to refresh my mind on the question. So, this is the preventive early on. I think the key thing there is that PET scanning. So the PET scanning can be done before you get symptoms. And this is one of the things, when I had a big table of all the antibodies that failed, one of the reasons that in the early trials, actually treating people they thought had Alzheimer's but didn't have it at all. And therefore, you know, these things were working because they probably didn't have Alzheimer's. Right. So now we've got these fantastic tests that are very specific. And so with these trials, like the lecanemab trials, they actually will treat people that've had the PET scan and either the sort of pre-symptomatic or what's called mild cognitive impairment people, and therefore the clinical trials are a lot more reliable than they used to be.
Yeah, thank you. We have another question for Chris. Chris, can you tell us some of your hopes for the future, particularly in terms of research and how research might be helpful for patients?
Yeah, just what they said just then.
Drugs that work.
That's basic, isn't it? You know.
Yeah.
How long will it be? You know.
Yeah.
I guess.
Yeah. Yeah, Michael, can you give any sort of idea how long does it take from for example identifying a target to actually taking it through the testing that's needed? For example, if you were to come up with a target tomorrow that was going to be successful and was then tested, how long would it take to show that that's a good target and that that's useful?
if you asked me 2 years ago, I'd say 15 years.
Right.
We do actually have 2 targets that are in that. We've identified 2, we've got structures of the 2. We're about to do the drug discovery on them. But you have to go through a whole bunch of things. You have to make sure that they're effective and in cells and safe in cells. Then you go to animals, and then animal models of disease, and then into human clinical trials. The great news with the lecanemab is it's already been going for quite a few years now. So it's in phase 3 with some pretty positive results. So there's a chance it might be out sometime next year, but that'll be up to the FDA. But it's 15 years. Now, why I said pre-COVID, it's something magical happened with COVID. There's some really bad things that happened with COVID, obviously, but there's some positive things. And 2 that I'll mention is, one is it brought a whole new technology to drug development, and that's the mRNA technology. Up until COVID, the FDA, other drug authorities around the world were too scared that new technology to actually accept it into human clinical trials. Because of the desperation of COVID, it was approved and done some miracles. The other thing is that all of a sudden clinical trials are a lot faster. So hopefully that it's not just for COVID, but hopefully all other clinical trials can be a lot faster now that the drug regulation authorities found ways of doing it.
Thanks, Michael. We've got another question. This is a slightly thorny one, and this is for Michael. But I think it's important that we talk about this. And this question is whether you have any comments on the recent controversies about fraud in Alzheimer's disease research, and how is this influencing the field, and how is it influencing your work? So your opinion on that, please.
Yeah, I think the influence is outside the field to the general public. But the first thing I'd say is, I think for us who are scientists here, that we do like to say we're actually just normal people, like everybody else.
Yeah.
So support football clubs, and cricket's coming up, and I'm really looking forward to it. And because of that, because we're just like everybody else, and probably lucky that we had the education to do something that's really enjoyable and makes a difference, is that we're a community like the general community. Unfortunately, there can be some bad people, lots of good people. And so this particular question about the fraud, what it's about is I presented the travel of the A-beta peptide going from the single unit through to these oligomers, multiple molecules of A-beta, through to the protofibrils and then fibrils and plaque. Between the oligomers and the protofibrils, this group published a paper in a very important journal, very high-profile journal, saying that there's a large oligomer that's responsible for the disease. So they characterized that, and they could show they could put it into animals and they could develop symptoms and so on. So, I was involved in the field when that came out, and all of us knew that there was pretty compelling evidence that these oligomers were toxic. All is said to us, or to me at least, was, well, here's another example just showing some pretty good proof that these oligomers are probably where you should be focusing on. It turns out that unfortunately it would appear that somebody did some fraudulent stuff, presented data that wasn't real. And the community has sort of come together and said, this is outrageous. It affects us all, puts a bad bad thing on us. But it hasn't affected the field because it wasn't as important to the scientists as people think. So everything I said today is not changed by that. I didn't mention that because obviously you don't mention that because it's now it's not something it means anything.
Thanks, Michael. That's really, really a helpful perspective to have. And I think the thing that I've remembered with this occurring is that science has a tendency to write itself. In the end, it's all about proving and reproving and showing that things really work. And eventually the truth is the thing that wins in the end with science. We've hit the top of the hour, and I can see there's still a few more questions to be asked, but we will answer those by email during this week. So don't worry, we will get to your questions. But I do want to honour people's time. Firstly, thank you. Thank you very much to Chris and to Kate. Thank you for being with Chris as well. Thank you, Michael, for sharing your insights with us. And thank you to all of you who've attended. If you'd like to watch the webinar again, or if you've got friends or family who'd like to see it, this has been recorded, and we'll send around a link in the coming days so that you can share that with others or watch it again yourselves. We're also going to be sending out a survey so that we can collect your feedback, and so we can keep bringing you events that you find engaging and helpful. And we'd love it if you would take just a few minutes to complete it. It's not going to be a long one. If you'd like to make a donation to support our research into Alzheimer's disease or of any of the other diseases that we work on here at SVI, please go to our website, which is www.svi.edu.au, and I will put that in the chat so you can find that easily. I'll just pop that in now. That's our website. Or contact one of the team at the foundation, and I'll put their email address also into the chat. If you're not already on our mailing list and you'd like to join it to receive information about our events or about our research, or if you just want to hear more about what SVI does, please copy that email address. There we go. Simone's also put that in. Please email Simone Flanagan at our foundation to hear more about it. But again, thank you both to Michael and to Chris for joining us and sharing your insights with us. I hope you've all really enjoyed this webinar and hope you have a good rest of the day. Thank you very much for your time.